Yeah, so I think the recording is switched on. So welcome everyone to this IEEE EMBS Talk Series. The first talk is going to be on oral cancer. The title of talk is going to be Tobacco Burden in Vulnerable and Marginalized Populations in Chennai, South India, a researcher's journey, and where engineers actually can pitch in. So we are delighted to mention that we have Dr. Sree Sucharitha. She is a founder, chief executive officer of Kairos Kinetic Private Limited. She was a former professor in Department of Community Medicine and research coordinator at Tagore Medical College and Hospital. She holds a fellowship in HIV medicine from ITech India, which is a joint program with collaboration with Tamil Nadu government. She's also a fellow of Asian Human Rights and Drug Policy. She's a founding director of AHRER, that is Association for Harm Reduction, Education and Research, a first ever registered medical professional body in India for increasing the public awareness of harm resulting due to various human behaviors associated with tobacco consumption, narcotics, alcohol, and obesity. So I give the stage to Dr. Sree. So this talk series is going to be focused on people who are in medical field like you, so that we, from engineers' point of view, try to understand where we can actually pitch in to help the medical practitioners as well as clinicians, um, and try to solve a problem which is going to be what is going to the aim of this talk. So
Yes.
thanks for giving an opportunity to join this session, Dr. Sree.
Yeah.
Yeah, so I think you can start now.
Yeah. Thank you, Dr. Uttam. That was a great introduction for first session launched today with this talk. Definitely, there is a more discussion towards the end, but towards the beginning of my presentation, I'll be sharing what are all the research I have undertaken in vulnerable and marginalized populations, as the title suggests, regarding tobacco use. And from there, we will pivot towards the session's major focus, which is about the collaboration between medical doctors and engineers in addressing healthcare burdens or healthcare issues which are of significant public health interest, such as oral cancer in India. So, when you talk about Kairos Kinetic, which is my startup, my team currently comprises my guide, Chief Technical Officer Professor Kannan, who is a microbiologist, immunologist of eminence, and he has deposited a bunch of new gene findings in CDC Atlanta from his work in immunology as well as microbiology. So, Sir is my guide as well as part of my team in Kairos Kinetic as Chief Technical Officer. Sir said he will join, but I hope he will join very soon. So, as per my introduction, these are my previous associations with Tagore Medical College and Hospital. I'm thankful for working with the institute for the past 10 years. Recently, before coming to US only, I submitted my resignation. Hopefully, they will accept and let me go to pursue my further interests and passions in my career. So, coming to the today's session agenda, I'll give a brief introduction because of your engineering background. I'll just lay out the foundation, how much tobacco burden is there in India in epidemic proportions, as well as that is the determinant for oral cancer being coming from Asia, especially from India, as one third burden is contributed from India. And I will list out some of my past research related to tobacco. I'm not sharing my other research here in today's presentation, though my interests are very diverse, including COVID-19, telehealth, artificial intelligence, in other areas. But today's session, we are only going to talk about tobacco related research and share some significant findings so that it will help engineers like all of you to understand there are so many problems related to tobacco and oral cancer. It is not unidimensional. Multiple presentations of oral cancer, multiple population groups expressing oral cancer is very unique. And what are the current research collaborations and what is the way forward? So, if you look at the tobacco epidemic in India, according to World Health Organization, 7.1 million premature deaths per year are attributed to tobacco use, and mostly they are coming from smoking, and this could reach to 8 million by 2030. That is hardly another 7 to 8 years. But the most interesting point from this first statement is majority of research has been focusing mostly on smoking part of tobacco, whereas in India in 2017 approximately 266.8 million adults in India have used tobacco in any of the forms, and out of this approximately 300 million adults using tobacco in India, 200 plus million are using tobacco chewing forms. That is of concern for us. When entire data is skewed towards finding smoking related tobacco mortality or morbidity, we are missing out of the picture the 200 plus million users using tobacco chewable forms. That is why I call it as iceberg phenomenon. This 270 million as per 2017 data is only the tip of the iceberg. Many forms of chewable forms of tobacco are not accounted in this 267 million. The maximum levels of smoking prevalence for both men and women in India, as you can see here, it is definitely on the higher end for men at 17.5%, comparatively 1.2% for women. As all of us know, smoking is still considered a taboo for women based on the gender dynamics. Societal determinants such as the weaker gender being the woman, and social taboos and social fabric deem that women should not smoke in India. Though this trend is changing currently, still 1.2% of women smoke against almost 17.5% of men. And when we talk about smoking forms, the most common form is not cigarette, but it is BDs because of the socioeconomic strata as well as the pricing index. Coming to the smokeless tobacco use prevalence, it is almost 19.28% for men and again 5.06% for women. And as you can see from here, smokeless tobacco form, which is like a discreet practice, allows women to consume tobacco more discreetly. And there is a social sanction because, according to certain cultural practices, when guests come to our home, loose tobacco leaves are offered. It is also consumed in the form of pan and other forms. So, smokeless form of tobacco used among women around 5.06% is again of concern because that is where cumulatively smokeless tobacco use with 200 plus million users and women and men equally using it, and more younger girl children exposed to these forms of tobacco at home because mothers, grandmothers are chewing tobacco. They are starting tobacco chewing as early as 7 years, 9 years as per latest research data. So these are all the dimensions we need to keep in mind when we think why oral cancer is becoming a prominent issue in India. So, coming to the other important aspect, unlike Western countries where there was a different pattern of tobacco epidemic, in India it is expressed as a compressed tobacco epidemic because the tobacco use currently is increasing among Indians at a time when the negative effects of tobacco consumption on health were already apparent. In the sense, like unlike Western countries, when the tobacco epidemic reached its peaks in 60s and 70s, 60 and 70, when tobacco companies had aggressive marketing campaigns targeting both men and women, claiming that tobacco has more benefits. It's Marlboro Men and it is torture freedom for women. Unlike that kind of scenario, right now we are almost aware that tobacco is a deadly product. It kills, it causes cancer in multiple organs in body. In spite of that, repeatedly my research also revealed that even in low income group populations like migrants, working class women, transgenders, everyone is aware of tobacco related ill effects on health. In spite of that, tobacco consumption is on the rise. That is why scientists like Rufi Sheikh et al. Have come out with this terminology called compressed tobacco epidemic, where the tobacco use is on the rise among Indians when already we are experiencing the negative effects such as cardiovascular disease, increased deaths from cancer resulting from oral cancer, etc. With this background, I'll tell you explicitly about oral cancer in India. As you can see here, one fourth of global incidence, that is number of new cases per year coming from oral cancer, one fourth of cases are from currently India, and it is going to be one third by 2030. And there are a variety of potentially malignant disorders in oral cavity, like inflammatory oral submucosa, fibrosis lesions, erythroplakia, leukoplakia, candidal leukoplakia, and congenital dyskeratosis, lichen planus. All of these lesions, when categorized under potentially malignant disorders, are preclinical phase of oral cancer. It is extremely important for us to understand when clients are presenting with this kind of picture, if you intervene and avoid the thing, there is no progression towards the cancer. But unfortunately, because of poor health literacy among the subjects, as well as lack of clinical timing in the OPD hours between clinicians and the clients coming to the contact in the healthcare system, this is where we are losing an opportunity, and thus leading to oral squamous cell carcinoma being 84 to 97% of oral cancer. That means it is the most common form of oral cancer for Indian subjects. If you look at the oral cancer scenario across India, it is not uniform. And this picture will help you to understand across India, different age groups, different profile of people between genders, and what are the common areas in the mouth, which are the most common risk factors for development of oral cancer, have been listed out. Take, for example, South, where we are in Chennai, the most common age at which oral cancer is found is 60 to 69 years. That is too late. By that time, already the person will be having either stage 3 or stage 4 cancer. Very much little we can do either through surgical remover or chemotherapy. That kind of options are only available. And most of these oral cancers are present in males, that is 61% of the times, and females are also coming up with almost 18.5% oral cancers are also reported from female populations in South India. And the common area of oral cancer for South Indian population seems to be the tongue. And common risk factors are betel quid, as we said, like in our cultural practices. Otherwise, also it's a common practice to eat betel quid with tobacco shavings, tobacco consumption in the form of cigarette, beedi, and chewable forms. Along with that, alcohol, non-vegetarian dietary practices, as well as poor oral hygiene. These are the reasons why oral cancer is co- uh, present presenting itself in South India. Now let us look at Northeast. When you look at Northeast, the picture is the average age at which oral cancer is diagnosed is 70 to 75 years. Again, very late. When people are presenting to a hospital or in medical institution or academic medical research institution at 70 and 75 years with oral cancer lesion, what can a doctor like me do? Or even if you engineers design a device and keep it there, what can I do with the device? So we need tools, early diagnosis tools, to take with us to go to the community, not to keep it in the institutions when people are waiting there to the patients so that they come at 70 years, 75 years. No, that's not going to help us. And in Northeast India, males are like 58.4% of the times having oral cancer, and shockingly female are 60.2%. Again, common area is tongue. Common risk factors: alcohol, betel quid, tobacco, and smokeless form of tobacco is most common there, and non-vegetarian. So, with these two comparisons between South India and Northeast India, I would like to draw your attention that the summary being oral cancer presentation across India is not uniform. It is varied. It is regional, and we need to go to that level of understanding to tackle the issue of oral cancer in India. Now, what is the problem in oral cancer detection? Because there are a lot of issues here other than tobacco consumption. Doctor patient ratio. There is a poor doctor patient ratio in India. We have 1.34 doctors per thousand population, which is one of the poorest ratios for doctor patient relationship across the globe. And as I currently explained to you, there are very many expressions of oral cancer lesions. Unless a trained professional is there, you cannot diagnose them in early stages. That is what is leading to late stage cancer detection. And when late stage cancer detection happens, there is low 5-year survival rates. That means from the point of diagnosis till the patient could complete another 5 years of survival, it is very low. Whereas if we diagnose them in the early stages, the 5-year survival rates improve drastically to 84% as against late stage diagnosis with only 10 to 12% survival rate. This is the real problem of oral cancer detection. Now, if you look at the media representing oral cancer things, this is the latest one on your left side from Free Press, which says keep an eye out for upsurge of head and neck cancers in India. This is from HCG Cancer Hospital. They say that tobacco usage is connected to more than 85% of head and neck malignancies. When they say head and neck, it includes oral cavity. Tobacco and alcohol use is linked to 80% of these cases. And the risk of this disease, that is head and neck cancers, is around 10 times higher in smokers than non-smokers. So in spite of all these pictures, as I said, because of concentrated tobacco epidemic, people still continue to use tobacco because of very many reasons. That is where we have to address this. And on the right side of your picture, there is a case, which is a high profile case, which made national news, where a 17-year-old boy from western state of Maharashtra was forced to undergo facial reconstruction surgery, 17-year-old, after developing a late stage oral cancer. He has been a gutka user from the age of 13 years. So again, I would like to draw the attention of engineers attending this. This is a very unique case study. At 13-year-olds, the boy started using gutka, the most common chewable form of tobacco available for low-income groups, from 5 rupees, 3 rupees, 15 rupees kind of pricing. Anyone can go and buy. The policies of non-sale of tobacco to minors is only on papers. It is not implemented strictly. It is available near schools. It is available near workplaces. It is available near anywhere in Chennai. Also, you can just try it, you can buy it. So from 13 years to 17 years, it's hardly 4 years, and he presented with late stage cancer. You just have to understand what could be the toxic profile inside that gutka packet for that escalated expression of cancer within 4 years. So the Dr. Chaturvedi from Tata Memorial says the hospital receives 80,000 new cancer patients every year, and out of which one quarter are neck and oral cancers, attributed largely to chewing tobacco. Again, the same thing which we discussed. No country sees as much misery and suffering from smokeless tobacco like India. For comparison, United Kingdom, oral cancers only constitute 2% of cancers diagnosed annually because they don't chew tobacco, right? Chewing tobacco is a practice more strongly in Asian countries and some Nordic countries. And in Nordic countries, that is Sweden and Norway countries, the smokeless form of tobacco they use is 99.999% safe compared to combustible forms of tobacco. That's why those countries have switched away from combustible to chewable forms of tobacco. So, coming to the crux of the problem in India, the rate of new cases being diagnosed has increased a staggering 68% in the past 2 decades. That's a good news because more people are getting diagnosed, but accessibility health services is low and poor health literacy because they're coming in late stages. So the advantage is nullified, and India is spending vast amounts of money in oral cancer treatment, and early cancer detection strategies can lead to 20% reduction in advanced stage disease, and it could save a lot of public health money. So, coming to the premise of today's talk, at present, the diagnosis is based on clinical exploration. I already expressed to you earlier that the very many clinical presentations of oral cancer makes it difficult for an ordinary clinician to diagnose them at early stages. You need specialized training, dedication, and compassion to engage with these clients when they attend your clinics or public health hospitals. So, the clinical exploration later forming part of any medical consultation, it has high discriminating capacity. Suppose 3 4 doctors are sitting in an OPD. I feel this is a cancerous lesion. My co-clinician may disagree, right? So these are all subjective interpretations. And in a clinic like India with primary healthcare centers loaded with outpatients, you can imagine, all of us are from India. We are not from other part of the world. We know how much of our primary healthcare centers are supposed to do. You have to do deliveries, you have to do immunizations, you have to do cancer screening. You just imagine the toll on the doctors. So that is where we are losing more and more opportunities in early detection of cancers. Now let me come quickly with that backdrop. What is my journey in tobacco as a researcher? Pre-COVID, I was doing this work very intensively with my interns, house surgeons posted in my department of community medicine. We went to all these construction sites. You are all aware, high-rise constructions, metro construction projects in Chennai. So we used to get permission along with my interns. I used to go there and conduct these migrant oral camps, and we used to do this health awareness on tobacco cessation, tobacco health effects to all these migrants. Because I'm the only Hindi speaking person there, I used to conduct these sessions, and used to give them medication, and we used to conduct their tobacco use survey: how much tobacco, what kind of products, how much duration they have been using. Because they are migrants, we used to capture from which part of India they were coming. And I used to train my students to conduct the oral cavity examination, and we used to take all these lesions pictures. After that, we used to take the oral swab using the brush methodology. We used to take a ice cream spoon, wooden spatula, and scrape the oral cavity whenever we found a client or a migrant with oral cavity lesion with consent. After explaining the procedure, we used to take the scrapings of the oral cavity and fix it on the slide. There was a nurse behind us, pathology nurse. And then you can see here, we used to do these things just in 2019 October. This was a year-long project where we reached out almost to 1,200, 1,300 migrants across Chennai. Very intensive 6 months project we were doing. These are all the lesions you can see here. The variety of or potentially malignant disorders, which I explained: erythroplakia, leukoplakia, fibrosis, you name it, you see it in their oral cavities. These are all the lesions from our own research, not from textbooks. So, with all these lesions and with all this work, we published this. A lot of work we have published defining the socio-economic environmental determinants for high tobacco consumption behaviors among interstate migrant construction workers. This was among the first batch with 345 migrants. And the conclusion was tobacco prevalence, especially smokeless tobacco, is alarmingly high in interstate migrant construction workers. The other study, based on our oral lesion screening, workplace based oral screening, and this conclusion also: prevalence of potentially malignant oral lesions using this tobacco is very high and it needs urgent interventions. And what are the social determinants? What are the socio-economic and cultural profile in access to tobacco prevention and support services? Are they getting anything? Conclusion is: reappraising the social determinants in access to tobacco prevention and cessation support services for migrant workers may be a promising strategy to reduce health harms. Just to give you a context here, being migrants, because of their working times and working hours, they cannot go to regular PHCs to attend for their health problems. So, if we want to address tobacco burden, we need to think of unique solutions like a migrant health program. Like in Kerala, Kerala has a migrant health program targeting uniquely for migrants. They have a special migrant ID card, and they are because of that migrant identification card, they are eligible for certain health services and certain other benefits. Similarly, if we think of certain health program at a national level, all these migrant related health issues can be covered. And tobacco is one of the major burden because they are staying away from the families. They think this is the only thing they can afford. They cannot afford alcohol every day, but they can afford 5 rupees, 3 rupees kaini gutka packets, and they keep sharing among themselves. And on Sundays, they get a cigarette. On weekends, they buy alcohol. This is the profile of a migrant lifestyle in Chennai who is coming from Jharkhand, Bihar, Bengal, and other parts like Uttar Pradesh, Madhya Pradesh, even from Orissa. So we need to understand all these socio-economic determinants, making them addicted to these products, as well as lack of services. For even if someone is motivated, they are not able to quit this, because the migrants are also coming with their families, with their wives and women. We try to understand what is their lived experience of tobacco use, and we also had a interview based study to understand. And among these 52 male and 5 female tobacco using migrants, the cultural acceptance and sanction for smokeless tobacco, peer influence on uptake of tobacco habits after arrival in the destination city, use of tobacco to overcome physical exertion and boredom were factors driving the higher use of tobacco among migrants. So this study reveals the complex interplay of socio-cultural economic factors associated with migration and disrupted social support system as contributing to high burden of tobacco consumption. So we can understand the interplay of multiple factors, and we also attempted to understand what is the urinary nicotine. As all of you know, people consume tobacco only for nicotine. Nicotine is the one which gives them the pleasure, and it is excreted as cotinine. So we wanted to measure how much cotinine is being excreted, because there is high consumption. Is it reflecting the same? And we measured this cotinine levels in urine among 78 migrant workers into three groups. We divided them into smoking tobacco, smokeless tobacco, and non-tobacco consumer groups. And the mean cotinine levels of urine for smoking and smokeless and non-tobacco users are displayed here. And the conclusion is analysis of variance showed that the urine cotinine levels for smokers are significantly higher than non-users, obviously. And similarly, smokeless tobacco consumers, smokers have increased cotinine levels similar to smoking tobacco. So in this particular small sample size study, cotinine levels are found to be very high. Generally, the acceptable levels of cotinine in a non-user are 10 grams, whereas here it is very much high, around 70, 80s like that. So it is very high. And to understand the interplay of tobacco among women, because I already explained to you, there is an increased trend of smoking among urban educated women in India. It is on the rise, especially in Chennai. So we wanted to understand what are the lived experiences of women tobacco users. And again, it's a qualitative explorative study where we interviewed 21 women tobacco users, different professions and marital status, graduates, postgraduates, how much duration they were using tobacco. Some of them were using tobacco for more than 5 years, 6 to 10 years, and more than 10 years also. This is a very complex picture, but if you can understand here, what are the reasons women are taking up of tobacco? Family problems and stress, thrill and curiosity, peer influence, family members already being smokers. These are the factors which helped them to consider initiating tobacco for the first time. And after the first time, whether they continued using tobacco or whether they quit tobacco, these kind of factors are very important for us to understand. If they continued uh, using tobacco, there are very many reasons, such as mental health issues, relationship crisis, and other issues. But if they quit tobacco after using for the first time or after using for a couple of years, the major reasons are they are getting health problems, medical conditions, being women, uh, polycystic ovarian disorders, thyroid conditions, mood swings, breathlessness, graying of the hair, and getting married. These are the reasons after taking up smoking, the woman has considered quitting and ultimately done with smoking because of these medical conditions. But if they continued smoking, what are the reasons? Because they are fine with the smoking habit. It gives them temporary pleasure. It gives them diversion from stress. They also consider themselves not being heavy smokers. That is a perception strongly prevailing among women smokers. And most of them are not aware of nicotine replacement therapy options for cessation of smoking. They are thinking with strong sheer determination and self willpower, yoga, breathing techniques, meditation techniques, I will be able to quit. But none of them have actually approached a clinician to help them quit tobacco because they think they are not serious smokers. Now, the other part of my tobacco work has been among LGBTQ, that is transgender community, and this paper has been recently accepted for publication and couple more have been submitted. The pattern of tobacco use and associated socio-demographic factors among transgenders living in Chennai city of Tamil Nadu. I kind of reached out to 934 LGBT. Spectrum is LGBTQ, but in this paper we tried to address only transgender population, which came to 734. And the result is the prevalence of current use of any form of tobacco products among transgenders. You could see it is almost 65%. So it is very high. It is three times higher than general population. You remember the smoking prevalence was around 18% among men, whereas here it is 64%. So any form of tobacco use currently in transgenders is 65%, and about only 27% have tried to stop using tobacco during the past 1 year. That is again concentrated tobacco epidemic, in the sense like in spite of knowing the negative ill effects of tobacco, people are still using and still continue to take up the habits. With that background, let us my research how helped me to start up my Kairos Kinetic company, because we felt that we are not having enough solutions to offer to this population when they are having so much problem. So I started my company Kairos Kinetic, which is a startup with the single goal of offering population health management solutions. It's not like I want to devise a solution, keep it for elite population like corporates, like keep it only for corporate sector. No, my goal is only to bring it to the population, like population health management solutions. So we started that, and this is my website. You can go and check it out. So with that first attempt, we applied for the government of India, and there is the seed fund scheme, and with multiple presentations to different panels. Finally, we succeeded with my guides, Dr. Kannan's guidance. We both appeared, and we addressed a lot of their questions and replied back with a lot of answers. Finally, we got a very small grant of 20 lakhs to work on this project, which is called Nano-Oral Kit, the dual lateral flow assay with gold nanoparticle for presumptive identification of oral cancer, because that has been my body of work for the past 6 years. We have seen the burden, we have seen the need, and we thought like we'll try to do some solution oriented work, and we are currently working on the prototype development of that. And when we look at the way forward, these are the interdisciplinary approaches. My interest has been in artificial intelligence, because a lot of efforts have been already taken globally to understand the early detection of oral cancer through various approaches in artificial intelligence. So, since 2 years, we are also trying to work with various scientists in Chennai, and also right now we have this collaborative proposal submitted uh, to ICMR through NIT Assam. Dr. Murugan is there, who is the PI, and I'm also one of the co-PI from. It is four institutions working together to submit this proposal. It is titled A Cost Effective System for Early Diagnosis of Oral Cancer through Histopathological Images, Autofluorescence Images, and White Light Images. A simulation, modeling, and visualization based on artificial intelligence principles. We submitted this proposal in February. Hopefully, I'm hoping very strongly and very deeply that we will get this grant and we'll be able to, because Assam and Northeast IIT there and here we are all collaborating. So it will be a very interesting project for us all to put our minds together and do something to address the oral cancer. Now, along with you, along with Dr. Uttam, we are uh, co-ideating on certain projects, especially Dr. Uttam is interested in proposing this. I hope it will pan out very quickly. We are also interested in developing some oral cancer optical image tools for early detection. And we have this other partner with us in artificial intelligence, Dr. Tarini, Professor HOD of EC Department in BS Abdul Rahman Crescent Institute of Science and Technology, where we have been ideating for quite some time for the past 1.5 years. And we are trying to work on this low cost smartphone based screening system, again using artificial intelligence principle to detect oral cancer. Some of the students are involved in this project. Before I came to US for this a brief 2 month break, I was having this discussion with the students who are currently in examination mode. But once I come back to India, we are going to reconnect again, and they are trying to build a artificial intelligence based screening system using smartphones, so that our healthcare workers, our interns can use the smartphone and screen the people attending the OPDs, our outreach camps. We can use this, screen them, and bring them back to the hospital if needed for further exploration. And this is one more international research we are exploring. The first author in this paper, exploring healthcare professionals' perceptions of artificial intelligence, Lucy Shinners. She's developing a questionnaire tool to assess the healthcare professionals' attitudes. I wrote to her quickly and invited her to our latest April 7th World Health Day conference at Tagore, which was an international hybrid conference I organized for the department and college. We invited her to speak on the COVID-19 pandemic and artificial intelligence, the role of AI in tackling COVID-19, as well as challenges in LMIC. She delivered an excellent hybrid talk. And we are exploring collaboration with her to do an all over India, what are the healthcare professionals' attitudes for the entry of artificial intelligence into healthcare industry as well as healthcare system. Currently in Tagore, we are doing a pilot study along with my co-author, Dr. Vaishnavi, the dental public health dentistry colleague, and our guide, Dr. Kanan. We are exploring in this mixed method study what are the perceptions of healthcare providers about artificial intelligence technologies. They are making very quick inroads. Are the healthcare providers ready? What are the attitude? That's where we are talking earlier. Unless we involve doctors from the beginning, there are a lot of misinformation and informational gaps. So this pilot study is going on right now, data analysis, and once this is over, we'll be rolling out full big study. And this manuscript is currently under peer review. Very soon it will come out. The early detection of oral cancer with OLO object classification using deep learning techniques, again, artificial intelligence paper, which we have been working with Dr. Varun Kumar, AI expert in SRM faculty in Chennai. And we have been working on this for the past 6 months, and finally it is going to be published very soon. And using our archival data in Tagore Dental College Hospital, we have mined it, cleaned it up. Along with that, on top of that, we have used open source oral cancer images. And Dr. Varun, using this image database, he has constructed this YOLO algorithm and he has brought out this kind of results. And we submitted this manuscript. And once it's out, I'll share with you all. So this has been our work in the past and current and future collaborations with engineers, artificial intelligence, as well as design engineers like Dr. Uttam and his team at IIIT and NIT Assam. And we are always open to exploring further collaborations with any like-minded discipline, because it's not just oral cancer, any health related topic, we are very much open either through Kairos Kinetic or just being scientists. Me and my guide, we are Dr. Kannan. If he has joined the session, we are very much open for all kinds of collaborations and knowledge sharing. And the way forward, as we are discussing, where engineers can pitch in, whenever an engineer is trying to address a biomedical health related issue, the first and foremost thing is you need to understand the sensitization of doctors regarding the product or the device or the unique solution you are mastering or hoping to develop. You need to come and sensitize them, like how Dr. Uttam left me a message in LinkedIn. I responded to him very delayed, but the moment we got in touch, we picked it up very fast. And we need to train doctors. Once the sensitization is over, we need to bring them to a formal structure of training, the repeated trainings, because in medical school, we have the CMEs called continuous medical education sessions. Now it has become much more rigorized because of COVID and Zoominars, and a lot of platforms are there. So we can train them about these interdisciplinary collaborations. And goal setting is very important. Ultimately, the tool or the device or solution, what is its goal? Where is it going to help? How the situation is being reversed or addressed? Unless we have these goal priorities, planning our research will, most of the times when we review literature, a lot of research is coming, but it is restricted to that single paper, single manuscript publication. After that, there is no use. Most of the review of literature I do, artificial intelligence, it's coming like so much of artificial intelligence is evolving, but it is restricted to papers. But nothing is available to doctors like us. We are struggling to address this problem. So planning research needs to address all these priorities, and that is the way forward. Now, with this presentation, I'll break now and go back to interaction, and we can always have more interaction now. And I thank Dr. Uttam and IIIT DM for giving me this wonderful opportunity.
Yeah, thanks, Sree. It was a wonderful presentation, especially the work that you have done, especially with the migrant people. Like these are the neglected people of of India, and we have a lot of migrant population in India, especially in West where I belong. We see many people who are from migrant coming from West, and here also we see a lot of migrant population, and also in transgender, again neglected population. So it's an excellent work that you have done. We really appreciate the efforts that you have put forward, and yeah, so that's really great. So I have a lot of questions, but I will wait for others if they have any questions. So yeah, so Gauri or Monisha or I think one more Ponmian. So if you have any questions, you can ask the question to the chat, or you can unmute yourself and ask the question.
Okay, so Uttam, I'll be asking few questions. Yeah. So thank you very much, Sri. It was a very interesting talk. So I have few questions. Shall I ask?
Please. Yeah.
Yes. So you were mentioning about quantification of the cotinine levels, right? So how
Yeah.
do you actually do what kind of assays? Just out of curiosity, I just want to know
Yeah.
what kind of
Yeah.
assays are there
Yeah,
for
it was
quantification.
a very small research grant I got, which is amounting to 50,000. So we could not do
Okay.
more there. But
Okay.
what we tried to do was we collected the urine samples
Okay.
from the migrants. We went to the construction camps. We took the permission of the managers there, and
Okay.
we collected the tobacco use history. And after completing the questionnaires, we instructed the migrants how to collect the urine sample, and we gave them the bottle with the full instructions, and they collected the urine sample and gave it to us. After
Okay.
that, we brought the samples under lab conditions. There are certain instructions to bring back the sample because it was summer. Then we
Okay.
came back, and we processed the samples in the lab immediately. That particular day we cannot do the experiment, so we have to pull all the samples. So we pulled all the samples, and at one particular point of the time, after we completed our sample size, my guide for that particular study was Professor Manikantan, who is a physiologist. So
Okay.
urinary cotinine assays, sir only performed, being the specialist in that. And
Okay.
sir has done one particular. I'll share the paper later.
Okay.
What is the methodology he has done? It is also
Okay.
right now under publication review. We submitted the paper. It's taking
Okay.
quite some time, but yeah, sir has performed the analysis. I was assisting him in the lab, and in
Okay.
Tagore Medical College only, we have conducted in the Central Research Lab where we processed the samples. We got the
Okay.
cotinine assay kits.
Okay.
Those are the reagents which are expensive, and we process the
Mhm.
samples. And, there are certain steps Sir has followed, being a physiologist and the expert there. He has
Okay.
performed the experiment. And after review of literature, as I had shared with you, non-tobacco users, the cotinine levels will generally be less than 10,
Okay.
kind of thing. But we observed that this batch of samples are having high things, so we repeated again to be more confident that we are not getting very high readings. Out of the entire batch running the samples, we took again another 20 samples to reconfirm our findings and rerun the experiment, and we got the same results. So it
Okay.
was like double validation of high cotinine levels. That is why your question is very important in another context also, not for scientists or researchers curiosity, because most of these global adult tobacco surveys, they go with a questionnaire. I'll come with a questionnaire and say like, sir or madam, do you use tobacco? Whatever they say, I will note it down and calculate it as prevalence. But all of us know when someone is having addictions, 50% of the times they don't admit they are having some kind of
Yes.
addiction
Yes.
like tobacco
True.
or
True.
alcohol. So
Yes.
when we use these surveys and think like in India tobacco prevalence is coming down, we have to match them with this kind of objective measurements. What is the
Mhm.
cotinine measurement, or blood cotinine measures, or serum cotinine measures, or urinary cotinine measures? If you match these two things, it is totally mismatching. That is what we argued in our presentation in Japan. This abstract was accepted for a Japan conference, and me and my guide, Professor Manikantan, we presented the paper, and we argued that paper-based surveys assessing the tobacco use prevalence in a high-burden country like in India should be matched with at least a subsample. If you are doing 86,000 survey and saying tobacco prevalence is coming down in India by 6 points, from 29% to 23%, 6 points decrease in tobacco prevalence, where is the objective measurements? Are you giving us cotinine readings which in the same population, subpopulation, saying the cotinine levels are stratified like this? You're not presenting that. So cotinine level measurement, though expensive, cannot be undertaken as of now, other than as research pursuit. I cannot offer it to my clients coming to the coming to the clinic. So these are all the barriers for cotinine assessment, cotinine usage as a regular biomarker for assessing tobacco or nicotine dependence among clients.
It's very interesting. Actually, I have, in collaboration with AIMS Delhi, we have developed certain biomarkers for brain trauma. So we find that over a period of time, certain biomolecules get degraded, right? So how is the stability of this cotinine? Is it like stable over a period of
Yes,
time, or?
yes. Cotinine compared to blood, because of non-invasive sample collection, we used urine sample because
Yeah.
convincing migrants to give a blood sample, it
Mhm.
was a challenging part. And at that point, we thought we'll try with urine sample. But if you look at the shelf life of cotinine, there was a definitive period like 72 hours, and that kind of period was there. So within that period, we performed the experiments.
Okay. And the concentration wise, any idea like how was the concentration of
Uh,
the
Con
cotinine?
Concentration wise, as I said, my guide was
Okay.
titrating all the reagents, and he
Okay.
said that all the parameters. I will share the paper with you.
Yeah, sure. Just out of curiosity,
Yeah.
wanted to know the sensitivity of the assay, like how it works, and
Yes,
that.
he conferred with some of his colleagues in US, because for us
Okay.
also it is the first time we are undertaking something beyond paper based to assess the tobacco dependence. He conferred with his colleagues in US, and he
Okay.
got some of the best agreement, and we proceeded with that because it was a research grant for us. It's not
Yes.
like we are doing on our own whims and fancies. So we
True.
got a small 50,000 grant. So we wanted to be utilizing
Mm.
that money and be accountable for our methodology.
Okay, interesting. And one more question, since like you are an expert in oral cancer, so what are the current drug targets that people are doing research on? For?
Exactly. This is one thing we are trying to explore because my guide, Dr. Kannan, is again an expert in molecular talking.
Okay.
Currently, there was this Azidas kind of pharmaceutical company next to our college. So one day we scheduled a meeting and went to them, asking
Okay.
them whether there are any novel molecules they are exploring for oral cancer. Can we be part of that novel clinical trials or anything? Unfortunately, they said they do not have. And we are
Okay.
exploring with another clinical trial consultancy whether any novel oral cancer drugs are coming into Indian market for clinical trials. I want to be part of that clinical trials as the PI or co-PI or
Mm.
in some, but unfortunately, so far we did not get the call. But novel
Okay.
drug molecules, I think we can schedule a call later because again, Sir is the expert in molecular docking,
Okay.
and we
Even
can
I have a lot of publications on molecular docking and simulations of protein drug complexes. So,
exactly.
out of curiosity, wanted to know
Yes,
what
because
what targets.
yes, I totally agree with you, doctor. Because recently, for one of the conference papers, I presented the molecular docking paper along with Dr. Kannan as guide.
Okay.
Azadirachta based, the plant based novel molecule. You might have
Okay.
heard about Azadirachta indica.
Yes,
Then,
yes, yes, yes.
yeah.
I have heard
So
about it.
there is one company in Chennai also from which globally a lot of scientists come to that company in Chennai to take their neem molecules for molecular docking for various cancer, not just oral cancer. So
Do they have any specific protein which they target, or it is going to be like?
again, I'll send you after this call that
Okay.
Azadirachta oral cancer abstract I presented in my January conference. I'll
Okay.
send you that abstract, and you and me and Kannan sir, if Dr. Uttam is interested, he can join too. We can have another call later, and we can
Yes.
explore that. That is one very novel area which we
Okay.
can because having such novel molecules, again, people are publishing because of that manuscript and that conference paper. When we are planning to submit that, we are reading literature,
Mhm.
majority of publishing, but it's not going beyond that. So at least now with this collaborations, we do something. We can definitely go to the trial level also.
Yes. And how do you think is the peptide designing with respect to oral cancer, how effective it is? Because it's all non-synthetic, right? So
Yeah.
peptide-based therapeutics, how do you see how much scope do you actually see it in context of oral cancer when
There
it
is
comes to
always
treatments?
scope for peptide molecules because I personally think this peptide structure is more friendly because oral cancer has its own mechanisms. I am
Yes.
more into this biomarker and genetic, but so far peptide structure based therapeutics I have not, to be honest, explored.
Okay.
But
Okay.
in general, if we apply the COVID principles, COVID therapeutic principles broadly, the cancer, what do you say? Cancer, you are more, are you more expert in cancer therapeutics?
Yes, yes, I have been designing molecules and peptides for cancer
So,
therapeutics.
so
Yes.
is there any unique difference between other cancer like breast cancers and oral cancers?
Yeah, there are different set of genes probably that gets overexpressed.
That's what we
So
are exploring in one of our current project, which is currently undergoing in the lab, which we are trying to explore the BAX gene and other genes for
that's
oral cancer.
okay.
And
Vax is for apoptosis, right? If I'm
yeah,
not wrong. Yeah.
yes, yes,
That's
yes,
true.
yes. It's for that. And currently, sir is completing the experiment. Before I came here, I'm just following that up. So
Okay.
we are, and also this current project which we got the grant is also based on biomarkers. So
Biomarkers.
we can expand this profile and go into that area also. But unfortunately, the peptide specific
Mhm.
structure based relation to oral cancer, I have to maybe read up a lot, or maybe you can take one session for us in the next
Yes.
October.
Yeah, yeah, sure. We'll definitely plan for one such.
Yeah,
Yeah.
definitely. I think there are a
Yeah.
lot of ideas which can come up, and we can write multiple proposals out of
Yes.
it, and we can start the trial. This is what
Yeah.
Dr.
Mm.
Sree is emphasizing, I guess. Rather
Yeah.
than doing work on only writing papers, we should start doing the trials. Try
Mm.
to
Yeah.
make devices and
Yeah.
start working. I think
Because
that is the
unless
more
it
important.
transforms into some kind of product which the people
Mhm.
in our country are able to be benefited from, I don't see the value of publishing those things and keep writing more grants. We
Yes,
can
yes.
True.
take one grant or one idea to its logical conclusions and let the people be benefited out of that one entire
Mm.
life cycle. At least one thing we complete, it will give our team a lot of satisfaction, because when I go and interact with them, they are looking towards us, asking, they call me like this, migrants call me, Dr. Sa, give me solution. I'm ready to quit this. Where is the solution? I can't buy this nicotine gum costing 100 rupees, but I can buy 5 rupees gutka. So what can I do? So if we can provide them some solution, and we can reverse this epidemic, only when you go to the masses, not sitting in corporate sector and offering genetics to hi-fi people.
Mm. True, true.
Yes,
Yes.
And there
definitely.
is so much of research we can actually, as doctor was mentioning regarding this genes and all, doctor, that is one unexplored area. We don't, because all the gene bank depositions are from US, and only 100 genes are deposited for oral cancer from India. Can you even believe that?
Yes.
Only 100 genes are kind of deposited from Indian oral cancer scenario, when you have one third global burden coming from India.
India itself. Okay.
So we can
Yes.
actually write for one genetic, because of your background also,
Mm.
we can, unless you find the gene specific targets, your
Mm.
docking, your drug
Mm.
target, even if you design the drug, even in the trial, we will not get results, no?
Yes, it's approach towards personalized medicine, right? So
Precision
we have to
medicine,
find
right?
first, yes, yes, you have to actually find out which kind of genes are overexpressed in a particular subset of population, instead of blindly giving a random molecule which might not work for person X. So it
Exactly.
should be like a precise one, as you have mentioned.
And
Yes.
in our, because we read a lot on this area because of our project and interest in Kerala, Trivandrum, everyone is doing wonderful research. They are finding new genetic markers for oral cancer from Indian population. Maybe in our next call, we'll pull out those papers, and using
Yes,
those genetic markers, we will do the molecular docking as you are proposing,
yes,
because those
yes.
are our Indian population genes.
Yeah, the chance that these molecules will work for Indian population is more likely because we are
Exactly.
we are hitting on a correct target.
Exactly.
Yeah.
Very few studies are finding Indian oral cancer gene markers from Trivandrum and other places. If you pull up those studies, which are really actually done on patient samples rather than theoretical and modeling experiments, we can take those and use these docking principles along with my guide and you and all of us, and we can identify what are those potential drug things. Then we can go to the next level.
Yes, true, true. We can do that. Yes.
Yes, that should be an interesting future potential work for us.
Yes.
Looking
Unless
forward
we
for that.
address this in multiple angles, oral cancer is like a real big menace right now. We need to bring all multiple angles, early diagnosis, tools, drugs, therapeutics, genetics, and everything. Because this has so far, and this entire skill set and tool set is available only to a niche market. I want to break it down and bring it to everyone possible, because at least IIT is taxpayers' money. I studied in government colleges. All of us studied with some kind of public funding schooling system. It's up to us to give it back to them also.
Yes, yes.
Exactly. Yes. That's true. Yeah. So, Dr. Gauri, I don't know if you would have. So I think it's the time for you to leave. It's already 5:30. So, you have any questions as of now, Dr. Gauri?
Mhm.
She was working in nanoparticles actually. So,
So I'm actually working on biosensors, and I'm very much excited to see someone talking about that and bringing it to the market and to the society. The talk was very interesting and also informative as well, ma'am. So even I work on developing like affordable or point of care biosensors. So that it could be used in a day-to-day basis, like one touch, how it has reached the home nowadays. So, but one challenge we look at here is like, as you mentioned, pertinent for identifying the tobacco usage. So, how much specific it is for identifying the tobacco usage? Does it have any interference or any interconnection with any other habitual habits or any other diseases? So the one challenge I feel here is like identifying the biomarker and its specificity. So how do
Exactly.
clinicians, because as a clinician, you know better than us, we take it like we'll take this biomarker and we'll evaluate it. So what is the challenge and how specific the biomarkers are? So
That's a very, very interesting and very important question, because as a clinician, even when you just go and ask the clinician to do a blood pressure measurement, we do the BP measurement in front of you. The reading will come. The reading will say 120/70, which is normal, 120/80, which is normal. Another person will say 140/80. But we will not just take the reading and put him as one thing or other. We'll take the entire case history, his family history, his lifestyle history, his stress, his current eating habits. So, in the entire perspective, we'll fit into this reading and we'll predict what is it. Is it temporary? Is it white coat hypertension? Just because he saw the doctor now, his blood pressure is elevated. So, this is like a quick scan, and we are trained in medical school to do this kind of healthcare delivery. Similarly, coming to your point, a biomarker is there. The specificity we will establish with all that kind of trials with sensitivity and specificity quotient and all that. But even when a biomarker is there, it will not stand on its own as a alone, single, lone wolf or lone tiger kind of scenario there. You have to fit it again in the context of that. Coming back to the quaternion thing, quaternion in the sample we have studied is so high, it cannot be attributed to anything else. And that's why we did the comparison between three people, non-smokers and smokers and other. But the bias there in that population is those non-smokers also came from the same migrant population. So as I said, they might have said they are non-smokers, but their urine is showing 40. The other people who admitted they are smokers, it is showing 70 and 80. So that is where we are telling, if we have a real good grant, we can design robust trials. We can do comparisons between migrants, non-migrants, homemakers. Homemakers will be good controls.
Okay.
Suppose in a home with no smoker, if there is no smoker or tobacco user in that family, that will be a good control. But even then, I'm a non-smoker, my husband is a non-smoker. We go every day to a bus stop, we get exposed to environmental tobacco smoke. Literature says environmental tobacco exposure, we can actually have nicotine levels excreted in our body. And diet, we eat tomatoes and other food products, we can excrete
Yes.
protein in our diet. So
Mm.
all these things, if you kind of nullify all these things and bring out, that will, as you correctly said, it will elevate the specificity, right? Even with all these factors, you have fit this value regarding the patient's or client's profile. What are other things? A plus B plus C plus D. But this specific biomarker, why we are doing biomarker based research, why research is so much marching towards this precision elements, that is one thing which will trigger the subject for lifestyle modification. I'm sitting in my clinic and forever telling this guy, quit tobacco, quit tobacco, quit tobacco. He's unable to do it. In spite of wanting to do it, he's unable to do it. But now you do this dipstick quotidian in front of him, you give this dipstick, go to the washroom, dip this and come back, and you say you are telling you have quit tobacco, but this reading shows that you are still using. So this will trigger them, this will nudge them, as Nobel laureates nudge theory in economics says, you have to nudge people. You cannot coerce people into any kind of permanent quitting behaviors. You have to nudge them, show them evidence how their life is being ruined by their own actions. If they lie to me, am I going home with heart disease? No. They are going to home with their own heart disease and cancer. So we have to nudge them by showing evidence. At that point, your cotinine, with whatever specificity and in the background of their history, lifestyle, and other things, if you fit that, that will help the patients very well in making this lifestyle modification, because unlike drugs and therapeutics, lifestyle modifications are the best, because you invest, even yesterday in that book they are mentioning, you invest $1,100 in the prevention, whereas you have to invest $150,000 in the name of treatment for cardiovascular disease. That's in US.
Mm.
So prevention makes sense. And in that prevention armamentarium, you have a biomarker or urinary cotinine or some other tool to nudge them.
Okay.
Interestingly, innovatively, because people are thinking these things are not available to us because we are poor, we are disempowered. You have to give them some empowering tools like this so that they feel empowered to take health as their own matter.
Okay. Mm.
These are the things we learn when we go to the field. The helplessness, they feel like, okay, I'm addicted, okay, I'm alcoholic, what else can I do? Am I do I have to wait till I die, or are you going to help me help myself? So in that gap, we have to offer them some novelty, which will help them a lot, because at the end of the day, all of us are human beings.
Mm. Mm.
It's just that we don't have money to buy that biomarker or genealogy test, whereas other people are buying and doing those changes.
Yes. Right. Right. Yes. And
So the
it's,
cotinine, if there is any possibility for us to bring down the cotinine kind of assessment, very less cost, and do it as a point of care doctor, we should explore that.
yes, ma'am. So, because I understood, like from your study, it is like, if it is like LFA based, so most probably immunoassay based. So, if we could work on some optoelectronics and bring that, it will be a very affordable thing. But again, the affordable thing, validation, since right now I could interact with a clinician who can evaluate much better than me. Because
I
every
will
time,
help you with any kind of clinical angle for any product you're developing, because I have good network of doctors in various fields.
yes, ma'am.
Dentists,
So,
we have cancer specialists, we have
mm,
gynecology, we have, if it is gynecological cancer, any field you are trying to design a product, you just talk to me once. I can find a doctor or professional related to that clinical department. We can go and get their buy-in in the initial stages. Process will become easier.
yes, yes, true, true. So, if I develop a product for detecting, say, for example, cotinine or any other cardiovascular biomarkers, so what kind of interpretation will doctors be familiar, or they will say okay for that? So, the kind of data interpretation, because if you take LFA, so it is like a qualitative, only the
Yes,
color change we will
yes,
observe. So if we could bring it to a quantitative level, say for example, if I have some threshold and fix the number, so for the doctors to have confidence on the developed product, so what kind of factors you will consider in such
yeah.
scenario?
Even for one Swedish grant, we submitted cardiovascular disease biomarkers project.
Mhm.
Uh, as you correctly said, for doctors to consider anything novel, as I said, suppose if you are trying to do this, because most of the clinicians in India, at least cardiovascular disease clinicians, are at least, if they are not young, less than 40 years, they may not be aware of this. But for that Indo-Swedish grant we wrote, we got a cardiovascular from US Mayo Clinic. He was already doing biomarker based research. So we got his inputs and submitted that grant. So my point in giving you these inputs is Indian doctors are not so well versed. So if you are trying to do something like this, we have to do some literature and print out a flyer like this.
Okay.
Very simple language. What is it all about? Then we have to go, as I put in my slide, we have to sensitize them first. Sense
Mhm.
what is that biomarker, what is that first you sensitize, they'll pick up easily, then they'll go and do their own reading.
Mhm.
Okay, then afterwards a couple of days later, 10-15 cardiologists or someone we can gather and do a small mini training or something. Then apart from that, we have to do literature review and see what parameters we are trying to see, how we want them to accept or promote or be involved in this. We have to define those criteria, Dr. Gauri.
Okay, okay, okay.
Okay, it is a researcher-led process rather than
Mhm.
clinician-led process here, but they will be engaged once they believe in the work and
Project.
read.
True,
With
true.
suppose if you are trying to develop a biomarker for, I don't know, for
For
example,
example, troponin. Troponin is
yeah,
a
troponin
biomarker
we
for
have.
C in cardiovascular, but we
Yes,
cannot restrict that. It is only expressed levels of troponin will be an indication of that. So, in at that point, I try to interact with few clinicians, but again, a point of care tool or device for cardiovascular diseases or for cancer, where the screening stage itself I want to address. So
so
at
that's
that.
where you have to take a multiple biomarker pool rather than a single thing.
Okay, okay.
That is why we went ahead even in our nanoral thing. We are not doing one biomarker, we are doing four.
Okay, so it's like a multiplex, just say.
Yes, yes. So even your thing, if troponin or XYZD, some
Mhm.
4 or 5 markers with good chances of predicting a cardiovascular event, if you can pool them and bring them together in one assay, the
Mhm.
chances of cardiologist listening to that will be better rather than a single thing. Because as I correctly explained,
Mhm.
one test will not fit alone. It has to fit into the clinical context, the background of certain other things also. So if you have a battery
Mhm.
of markers, a battery assay, a multiple assay, 5-10 markers at one go kind of thing, that will help
Mhm.
them to believe like, okay, multiple things are being known here. So multiple, and you assign a risk score to each of these, and cumulatively, at the end of which you have 100 out of 100, 10 markers are giving a cumulative score of 90 or
Okay,
60,
okay,
your score is low, moderate, maximum. That kind of interesting profile will help them to accept.
okay, okay. Mhm. So that's actually correct. So I will work on these identifying the markers and developing these point of care tools. So maybe at some time later, if based on your availability, we'll have
I am available anytime.
yeah. So all three of us are we can think of some proposal where, because for CB there's no screening tool as such. Even troponin is also, it's in the later stage. So I
Exactly.
cannot claim that is like a point of care where it could avoid. So such kind of what to say inputs from the clinician will help us to devise a better tool.
Exactly. And as I suggested, you have to go with all these parameters to clinical history, family history, and the lifestyle habits, and all these
Yes.
things, plus this. So within that context, if this is coming in that place, it should not come as a standalone single agent marker with
Right.
multiple and as a risk score assigned. So with all this innovative, complex thinking, we can definitely go forward, because now this is the future way the science is evolving. There is scope for these things.
Right, right, ma'am. And also from our research side, we are working on enhancing the sensitivity or enhancing the limit of detection by working with nanoparticles, LSPR,
Mm,
PRS. So those techniques I could implement, but this
ye-
clinical side, we could get your inputs, which will be very, very much helpful for us.
yes, yes.
Thanks a lot, man. Thanks a lot for your time.
Yeah, thanks, Dr. Gauri. So I have just one small question, that is, to understand, as you said that even a small boy of around 13 years, he started to have smokeless tobacco, and in only 4 years, he was having this oral cancer. So I want to know that this smokeless versus smoke through smoke. So is smokeless version is like more malignant in as compared to smoke, or like?
That is the situation in Asian countries, Dr. Uttam. The smokeless form of tobacco, as commonly consumed, like kani, pan parag, gutka, and all these things, they are kind of having a lot of, as per some of the research from India, it is having 7,000 chemicals, out of which at least 100 are known carcinogens.
Mhm.
Okay.
I see.
And there is a lot of body of research about Indian smokeless tobacco products, and unfortunately we don't know what goes inside that. Okay, they
Mhm.
only sell it's a tobacco product, but a lot of chemicals go inside that, and that is what makes it toxic. And I shared the example of Nordic countries where they have this product called snus, Swedish snus, which is like 99.999% safe, because from 2000 onwards this product has come into the market in Sweden, and from now to almost 20 years later, we have a body of research saying that the country has shifted from smoking to this smokeless form of tobacco consumption, because smoking is causing lung cancers there. They transitioned to this, and in this 20 years, through pasteurization efforts, they have removed these tobacco specific nitrosamines. These are the most common culprits for the carcinogens, and they removed this tobacco specific nitrosamines to such an extent. Now in 2022, the Swedish snus products are 99.999% safer than cigarettes. And if you go and check out the literature, female smoking rates have historically fallen down in those countries because they are transitioning to this smokeless form of tobacco called snus.
Mhm.
And oral cancers, lip cancers, throat cancers, all these cancer incidences over 20 years, literature has come down to share that because of this pasteurization procedures and removal of tobacco specific nitrosamines, it has been made safe. So, unlike that form of smokeless tobacco in India, there is no such thing. And certain lab analysis of current market available smokeless forms of tobacco revealed 7,000 plus chemicals, and 1,100 of them at least known to be carcinogenic.
Mhm.
This is the reason why our population, that is what I was telling to doctors earlier, when people deposited oral cancer genes in gene bank, only 100 genes were deposited from Indian population. It is very less, and we cannot compare that because all these chemicals, tobacco plus alcohol will trigger a different combination, tobacco plus alcohol plus weed. And in this low income population, from my study, in the slum population, they are using multiple substance abuse. They are using marijuana, they are using weed, they are using alcohol, they are using cigarette, they are using bd, they are chewing tobacco, what not. It's like a potpourri there with multiple toxins going on there, mixing in whatever combination, whatever duration, whatever poor nutrition, poor oral hygiene, and all these immunocompromised states. That is what I'm telling. The research is lacking to really understand this dynamic between these socioeconomic groups. That's why I typically focus my research on this vulnerable population, because no one is understanding their reality, but people are telling they are addicted. But why?
Yes.
Even
So
these
maybe.
migrants, even these migrants, they are using tobacco. Women are eating tobacco because to suppress hunger, because they don't have money to eat. When they are working, they don't want to feel hunger. They keep on chewing that gutka pan parak so that they don't feel hunger. Till night time, they don't have a meal.
I
So
see.
poverty is also one of the reason.
Yeah, like the sad
And
part of. Yeah.
that 13-year-old boy, 17 years, coming with a fatal form of oral cancer, and this entire cheek has to be cut off because of that cancer, and facial reconstruction has to be done. That was one case in Tata Memorial, which made headlines because of the very young age. Again, the kid is telling he started using from 13 years, but data is telling from India the age of initiation of smokeless tobacco for girl child is 7 years. But boys, they don't use smokeless tobacco. They straight away start with smoking product because of that male factor, heroism, and gender role. And they are starting with smoking at 9 years.
Mhm.
Whereas girls are chewing because dadis and mothers are chewing tobacco. It is like that they start chewing tobacco from 7 years. And we have seen women, oral cancer patients lying in cancer wards. After 1 week of addressing their problem, again they will crave for that. They will beg, cry their family members to bring that candy gutka packet. Even if I'm dying with cancer, let it be. Give me that kaini. So
Mhm.
these are all the real things. But smokeless tobacco in India is real toxic bouquet.
Mhm. I see. That's great. So that's great information that we all three of us actually received from you, and actually that is the what is the requirement as of now. That is because we are unaware of the actual perspective that a medical person like you have, and I hope that we can follow up this discussion with real action, as you were saying, come up with trial cases, write proposals, and instead of writing only papers, we focus on conducting trials of our instruments for a large cohort of patients, and then come up with of the results. And once we come up with
Exactly.
early diagnosis, we come up with drugs also, which are very cheap, with which we can share with the person who is suffering and lower the suffering.
Exactly. That should be our ultimate goal with all these collaborations and knowledge sharing. If we can address this and try to dent it through our collective efforts, that will be a very good contribution from all of us.
Yes, definitely. We look forward to that. So, any more questions, Monisha or Gauri? You have any questions, or that's it from our side?
Yeah, that's it. As you mentioned, we will keep a follow up and try to do
Yes.
actions
Yeah,
towards it.
we'll work on some proposals and projects which will be more specific, targeting the oral cancer. So let us also, as ma'am mentioned, I'll also check for what are the molecular drug targets for the oral cancer. I'll look on the literature, and I think we can also do some more work on that, like molecular docking simulations, in vitro
Yes,
assays for drug development. So we can
yes,
also
doc.
look at the peptide based thing also we can do, and how that can be used as a marker for diagnosis. Those things also we can actually work with.
Because
That's
not much is being done in this area. Anything we propose, we can actually start small as a cohort, small cohort pilot studies, and immediately go into field level rather than restrict with paper publication and moving to the next thing.
yes.
We can go to the field level. If you are proposing peptide thing or anything, we'll definitely go to the designing of the drug or something with the collaboration with the industry, and then we'll go and try to do the entire gamut of the things. We'll complete one life cycle with one ideation that should give us a very good confidence to bring
Okay.
it out.
And one more question. So, do we have in vitro facilities for doing experiments at Tagore? Like, do you know anywhere we can do the in vitro assays part? Because
Yes,
currently we are building on the labs, but right now we have to do things. So, is
exactly.
it possible for us to collaborate for the facilities, like for
It
doing in vitro assays?
As I was telling with Dr. Uttam, it's not just Tagore. The world is so much better now with so much of things opening up in Chennai itself because of this COVID. A lot of people have started their labs for in vitro, this and that. You just get the things ready, doctor. I will
Okay.
help find where we can go and get the in vitro
Okay.
or in vivo or trials. Everything
Okay.
is possible. We have our own contacts and peers and networking. We'll
Okay.
find the way and we will do it. That's it.
Okay. Okay. Thank you. Thank you very much for your support,
Yeah,
Dr.
we do it because people are able to do things in garage and go to the next level. All of you are scientists with so much of brain
Yeah.
power. We can do much more better.
Yes, 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 we can do it. Yes.
Yeah.
Mm.
Yeah. So, thanks a lot for the encouragement words. Those are really required for us to like proceed forward. So, That's it from our side, and thanks a lot again for the talk. As we've recorded, we will by tomorrow or so we will share the recording perhaps in YouTube, and we will share the recording also so that you have note with yourself also.
Yes,
Thanks a
thanks.
lot. Yeah, thanks a lot,
All of
Dr.
you
Sree.
keep safe and we'll keep working together. Anytime, please drop me a mail. I'll be available anytime with 1 or 2 days adjustments probably. But otherwise, I'll be available, and most probably I'll be back in India in July. So I'll come and drop by the institution too.
Yeah,
We can have more in-person brainstorming sessions, and we can do much better.
yeah, that is great.
Thank you. Thank you.
Thank you all. Bye bye.
Thank you.
Thanks.
Thank you. Bye-bye.